Thank you all for coming. We're delighted to have you. It's going to be a great night. So resilience, empathy, and plasticity are all important parts of the human experience. But how do these traits actually emerge biologically? Renowned scientists, Dr. Dorothy Kozlowski, Dr. Peggy Mason, Dr. Janice Urban will come together to discuss their groundbreaking work in neuroscience. Discussions will focus on the biology of empathy and helping behavior in rats, the incredible ability of the brain to repair itself following traumatic injury, and how the brain develops a stress response and resilience. So our first speaker is going to be Dr. Janice Urban. Dr. Janice Urban earned her BS in psychology from St. Mary's College, Notre Dame, Indiana, and her doctoral degree from Loyola University's Stritch School of Medicine in the Department of Pharmacology and Experimental Therapeutics. Dr. Urban completed postdoctoral fellowships at the University of Washington and Northwestern University. She then became a research assistant professor at Northwestern University in the Department of Neurobiology and Physiology. She has advanced from an assistant professor in the Chicago Medical School of Finch University Health Sciences, which is currently Rosalind Franklin University of Medicine and Science, to professor and chair of the Department of Physiology and Biophysics. She is currently the director for the Center of the Neurobiology of Stress Resilience and Psychiatric Disorders. Dr. Urban's current research program focuses on neural processes that control stress response and the development of resilience. Her work also extends to the complementary collaborative projects on traumatic brain injury and the coincidence of anxiety-related disorders. In addition to her research, Dr. Urban is involved in various mentoring activities within the university. She has served on study sections for the National Institutes of Health and National Science Foundation. As an active member of the American Physiological Society, she has served on the advisory committee for the Central Nervous System section, awards committee, and currently sits on the education committee. More recently, she served as president for the Association of Chairs of the Departments of Physiology in 2017 and 2018. So I'm thinking all three of our speakers should come to the panel or just afterwards. Okay, so why don't we welcome right now Dr. Urban.
Thank you very much for the introduction, and I want to thank the organizers, particularly David, for the invitation to be on this panel with Drs. Mason and Kozlowski. I also want to thank the SNOOC, SNUC Society, as well as C2ST and DePaul for sponsoring this event. I'm looking forward to a very enjoyable evening. So I'm talking to you today about resilience. And I thought about what should I put up on the first slide? And I thought the fact that we had to cancel the our first session because of weather, and I thought the crocus coming through the snow is a perfect representation of our resilience through this winter, and the panelists to be here and all of you to be here today. So before I start to talk about resilience, I want to talk a little bit about a stress response. And right now, as I say the word stress, each of you is coming up with an idea of how you identify stress. Physiologically, when someone's walking down the street, so this person here, and all of a sudden a mugger comes out and he's got a knife. So almost instantaneously, when this person sees that knife and that person, the hypothalamus in the brain becomes active, and it stimulates the release of corticotropin releasing factor. This in turn circulates to the pituitary to stimulate ACTH or adrenocorticotropin hormone, and then we get activation, it circulates in the blood to activate the release of cortisol from the adrenal, and we also get adrenaline or catecholamines released from the adrenal gland as well. So together we have cortisol and adrenaline are ready to mobilize. Yes. Oh. So we have cortisol, which is a glucocorticoid, which provides energy for eliciting a stress response, and the adrenaline, or the norepinephrine, that causes the increase in heart rate and blood pressure. So all of a sudden, almost instantaneously, the endocrine system is ready to go to fight or flight, depending on what the threat is. And I would be remiss to not mention that once we have the endocrine response, the brain needs to take over and decide, is this a threat? And if it is a significant threat, what are we going to do? And so some of the brain regions that are involved in that are the cingulate cortex, the hippocampus, and the amygdala. And particularly, I'm going to talk to you today about the role of the amygdala in stress and resilience. And what I want you to know is that this brain region of the amygdala, it is involved in emotional reactions, which is fear responses. And similarly, as it elicits a stress response or a stress behavior, stress itself can regulate the activity of this brain region. And that's where we start getting into problems with stress-related disorders and anxiety. So a stress response is actually a good thing. I mean, so when you think about it, phylogenetically, when we have a zebra, munching on grass in the savannah, a lion comes out, boom, we've got the same endocrine response. The zebra knows that this is a bad thing and we need to flee. And so it runs away, and then it saves its life. And so that's an acute response. And that's a good thing. And a lot of you have, experienced an acute response. It happens and it's over. But what happens is that in our life today, we have the emergence of chronic stressors. And I think many of you can identify with this person up here, in that the stress responses that we have come to know as being acute become chronic, where we're multitasking, we're dealing with money issues and deadlines and exams and grant deadlines. And so this acute effect of a stress becomes chronic. We have populations of our soldiers and our troops who, when they go over either in peacekeeping missions or war, they're 24/7 living in a stressful situation. And people who have long-term diseases are also involved. This is a long-term stress. And so that has significant effects on the brain. And so when people are undergoing chronic stress, typically what happens when it's unchecked is like the brain regions become hyperactive. When they get hit with so much stress, they don't know when to shut off. And here we look at an MRI. It's an imaging of the brain. And this little orange area here is the activity of the amygdala in a patient who's healthy, who has no known stress disorders. Below here, we see an MRI of the amygdala from someone who has post-traumatic stress disorder. And so what we see is that the area, this orange area, which indicates activity, is much larger and much brighter, indicating that in these people who have PTSD, the amygdala is hyperactive. And that's not a good thing, because when you have a hyperactive amygdala, what that does is that increases your vulnerability to get a stress-related disorder such as anxiety, major depression, or post-traumatic stress disorder. And some of the numbers that we see, I just want to make a point here, is that while 3.5% of Americans have PTSD, what we're looking at is those troops that come over from multiple tours of duty, whereas 20 years ago, these war veterans would have an incidence of PTSD of about maybe 10%. Now, because they have multiple tours of duty, we're seeing increased PTSD in this population upwards to 20 to 25%. So this is a growing concern. And when we look at all these numbers, they look pretty big. And if you consider the percent in the population, you can figure that approximately 15 to 20% of the population at one point in their life is going to have a stress-related disorder. That's pretty bad. But when you think about it and you flip it around, that means 80% of us, 80 to 85%, are resilient. That means that we're able to take this stress and handle it well. We're able to adapt well in the face of adversity and significant sources of stress. And so resilience, you can kind of think of it as an emotional bouncing back. You're going about life, you've got an exam, get stressed, you take the exam, and then you're pretty much back to a baseline, and it doesn't exacerbate. So this is resilience. And so my lab has been interested in trying to focus on, well, what causes resilience? What is it about the human brain, or what is it about the brain that makes one resilient? We have tons of studies, some of my own, which look at how stress affects the brain. What we want to know is, what are some of the mechanisms, what are some of the factors in the amygdala that promote resilience? How does this mechanism work? What brain circuits might modulate stress and promote resilience? And most importantly, how can we increase resilience? And that's a big one. So to start, we took a look at the amygdala, and I'm calling it the basolateral amygdala. It's a portion of the amygdala nucleus that's called the BLA. And so I think of the amygdala, we talked about it being a fear, being important in fear, and I think of it as an anxiety stat for behavior, in that when you have a stressful situation in the amygdala, when you're anxious, you have these excitatory amino acids, glutamate, and we have the stress hormone, CRF, that is released into the amygdala. You get the fear response, and you deal with the fear. But what happens is that this is not unchecked. And so once we have a release of these excitatory factors, on the flip side of this fulcrum, we see that there are a number of compounds in the brain, serotonin, GABA, and neuropeptide Y, that act as stress buffers to decrease anxiety. And I indicated here that serotonin and GABA are already targets for some of these pharmaceutical agents that you know as Prozac and Valium. And so we have been focusing primarily on neuropeptide Y as a novel mechanism that might be something novel to target in addition to some of these anti-anxiety compounds that might give us a new way of thinking about how stress and resilience is handled in the brain. So what do we know about neuropeptide Y? Well, in a number of animal studies, it reduces anxiety, and I'll show you some data in a little bit. And that when we look at either the cerebrospinal fluid, the fluid that surrounds the brain, or in the blood of patients with PTSD or depression and suicide attempts, we see that they tend to have low levels of NPY. And so low levels of NPY tend to correlate with the development of some of these stress-related illnesses. There was an interesting study that was done a number of years ago where they looked at the Special Forces combat soldiers. Now, these are the people that, are like the SEAL team. They're just really very resilient, and they're seeking out the higher risk tasks. And when they look at their blood, they see higher levels of NPY. And so we have a lot of correlational evidence in humans that suggests that NPY, having high levels of NPY, is actually a pretty good thing. So we started to work in an animal model, and this is some work that was done by a collaborator in Indiana, Tammy Sajak and Nantha Shaker's lab, and we looked at this model of NPY-induced stress resilience. And so what we were able to do is we can take a rat, we used male rats for this, and we can discretely inject our neuropeptide Y, NPY, directly into the BLA. And what she did was she did daily injections for 5 days, and then she looked and monitored social interaction at various time points of these injections. Now, the social interaction here, this is a test that's pretty easy. You can see 2 animals here that are socially interacting. We take 2 animals, they're both males. We put them in the arena. So we'll put one in one corner and the other on the other. And then we monitor and check how much time they spend interacting. And this is actually an indication. This has been a long standing test that's been important for assessing anxiolytic compounds, and that they find that the more time animals spend interacting is associated with a less stressed or anxious phenotype. To put that into a context that some of you can understand, if you know you have a test coming up, you know you're not going to be socially interacting with whatever, going to sporting events or bars, so that your social interaction time when you've got a test coming up is going to be decreased. So when we look at the behavior of these animals, so we see, I want you to focus on the blue bar, which is neuropeptide Y, NPY. And so we're looking at social interaction time, change of baseline, and we're looking at 1 day 5, week 2, 4, and 8. And so now remember we have injections on day 1, 2, 3, 4, 5, and so we see an increase in social interaction, which is great. That means that there's more social interactions, the animals are less anxious. And we expected to see at day 1 and day 5. But what we notice is that SI time, social interaction, is elevated 2, 4, and 8 weeks past the injections of MPY. Now, what this means is that, well, MPY increases social interaction, but it also does something much more amazing in that these local injections, which are constrained to this time point, are causing changes in behavior way out to two months post-injection. So this gets at kind of a plasticity too, but I'm not talking about plasticity. Dorothy is. So that increase. So we have a decrease in anxiety. What about resilience? Well, so what Tammy did then is we actually injected the animals with MPY just like we did before. Day 8, so we looked post the injection time. We put the animals in a restraining tube, so it's a little plastic tube that they sit in there and they're restrained. They don't like it too much. It doesn't really hurt them. They just don't like being restrained. 30 minutes later, we put them in the social interaction arena. And what we see over here on the right side, if we look at those animals that were treated with the vehicle, that when they're stressed, we see a decrease in social interaction time, both at the day 8 and week 8. So that's pretty much what the normal response we'd expect to see. However, when we take these animals that are treated with MPY, and then we stress them and look at their behavior doesn't change from their baseline. So what this suggests is that these animals are behaviorally resilient. So that, a behavior that should be normal for them, a social interaction, is not affected by a stressor, which is not really life-threatening to them. So this is our model of MPY resilience. So the next question we wanted to know is, how does MPY work? And this I collaborated with Bill Colmers up in Alberta. 2 minutes? Oh. Oh, okay. And so what we found was that we identified that when we actually put MPY on, it decreases the activity of BLA neurons, which is perfect for what we would expect an anti-anxiety compound to do. And we identified that it actually works through this H current, which is a particular channel that is relatively specific to the amygdala. And when we look at our model of stress resilience, that if after we give MPY, we notice that this channel is actually decreased by NPY in the long term. So we think that through through MPY, it acts through this H current to decrease anxiety, and we also think that it is important for the induction and maintenance of stress resilience. So to see if this channel was really important, what my student Maria did, is we had a molecular tool where we could knock down this H current channel. And so by giving a virus that had a construct that was antisense for the H channel, we could inject that into the BLA and then see if we knock down this channel, is this going to reproduce what NPY does? And so we have our controls here where we actually could knock down this process, knock down the channel to about 50%. And when we look at the behavior, what we see is that, oh my gosh, when we knock down this channel, it looks just like what happens with MPY, that we see an increase in social interaction after 2 time points, and that if we stress the animals, again, we have a control animal where there's 50%, we have a decrease in social interaction, but once we knock out that channel, that current, that actually enhances and produces our stress resilience. And so what we think is happening here is that when we have a stressor, we've got activation of CRF, which activates this HCN channel, which which causes a stress response, and then protracted after about 15 minutes to 30 minutes after the stress, we have the stress buffer, NPY, that comes on to decrease the activity of these channels. And so we think that this interaction in this cell is very important for stress resilience and social interactions. So now the question is, how can we increase resilience? And so do the moms have it, is what I want to know. And so there's literature out there indicating that both in humans and in a number of rodent models, that females who have experienced reproductive processes, such as the process of pregnancy and rearing children or pups or litters, that they tend to have better stress responses. And so we did a recent study where we took virgin non-mated females or mated females. They went through a pregnancy, weaning, and then we looked at social interaction, behavior, and then we did the stress resilience test. And what we see is, yes, the moms do have it, in that apparently that reproductive experience or maternal experience, while it doesn't necessarily affect the baseline social interaction, what the important thing is that when we stress the virgin females, we see a decrease in social interaction, indicating that they're pretty stressed. But that these moms, once they, when they're in the restraining tube and then they have social interaction, they don't have that decrease in social interaction, that in fact this is a behavioral resilience. And so in summary, what my work has showed is that NPY decreases stress and anxiety and promotes a novel model of resilience through modulation of a particular current. And what we're thinking is that we're hoping that this novel interaction between a neuropeptide and a current might be some new novel drug target that might be able to help with stress illnesses. Finally, we've been able to show that experience can increase resilience. Now that we've been doing this with moms, we're actually going to be doing some paternity behaviors in rodents, as well as looking at exercise and some other behaviors too. And then finally, by looking at social interaction, it gives us a good readout of what the animal is feeling as far as anxiety. But this model is very important because social interactions within humans is very important coping mechanism for stress. It's been very useful in helping treating patients with PTSD, and also increased social interaction has been very useful in having good outcomes in improved aging in the elderly. And then finally, the we that I've been referring to are a number of these people here. My student Maria is responsible for most of the work. She just went off to Columbia University. And Megan Gillum is a graduate of DePaul from a couple years ago. So I thank you very much for your attention.
So thank you very much, Dr. Urban, for that very interesting talk on resilience. And our next focus is going to be on empathy. And I have the delight to introduce Dr. Peggy Mason. She received her BA in biology in 1983 and her PhD in neuroscience in 1987, both from Harvard. After postdoctoral work at the University of California, San Francisco, she joined the faculty at the University of Chicago in 1992, where she is now a professor of neurobiology. For 25 years, Dr. Mason has worked on the cellular mechanisms of pain modulation. Since 2008, she has pioneered work on the biology of empathy and helping behavior. Dr. Mason is committed to increasing interest in neurobiology through teaching. She taught medical students for 25 years, during which time she wrote a textbook, Medical Neurobiology. And Dr. Mason is currently the inaugural director of the undergraduate major on neuroscience at the University of Chicago. She also teaches a popular course, Coursera course that has attracted almost 175,000 students to date and has received multiple awards for her teaching. So please join me in welcoming Dr. Peggy Mason.
Thanks for coming out on a cold night. Okay, so we are going to talk about empathy and helping. So this is, the first thing I want to say is that I was minding my own business doing research on how morphine blocks pain, the cellular mechanisms of how morphine works. When this young woman here, Inbal Ben-Ami Bartal, who was a graduate student at the time, came and said, I'd like to look at the biological basis of empathy. So this entire thing was the idea of this entire line of research was the idea of a graduate student. We collaborated for years until she went off to do a postdoc at Berkeley, and now she's starting her own lab at Tel Aviv University. So that's to tell you students that you can do that. You can have your own idea and follow it through, and see it come to fruition. So what I'm going to tell you about today are the good, the bad, and the ugly. And I'm going to actually come out against empathy, okay? There's a trick. And so I'm going to just tell you what I'm going to tell you. First, what's the good? It feels good. Empathy, helping feels good. So that's the good part. The bad part is that it takes some resources. So it's going to deplete you. And the ugly part is that you can fool yourself into thinking you're helping when you're not. Okay, so when we started this, I mean, obviously you know the answer to this, or else I wouldn't be here. When we started this work, the question that Inbal wanted to look at was, is there a biological basis for empathy? Or do you act empathically because because of your schooling, because of your upbringing, because of culture? And so the idea is, well, if a rat shows helping behavior, empathic helping behavior, then this is not something that you're taught or something that you learn. It's something that is, part of your biological inheritance. And so to ask the question whether a rat would actually help, we did a very simple experiment. Our experiment is simple. As one of our colleagues said, the experiment could basically have been done in the 17th century, except they didn't have acrylic back then. But they could have used glass. So in the experiment, we have an arena that's about yay big, and in the center is an acrylic tube, and a rat is put into that acrylic tube, and that has a door, and the door can only be opened from the outside. So the rat is trapped in the acrylic tube. And another rat, minding his own business, can continue to mind his own business, or can decide that it's worth his while to go and open that door. We test the animals repeatedly for 12 days. And if a rat opens a door, it's actually no big deal. What you really want to see is whether the rat opens the door again, and then again, and does it very quickly. Well, as it turns out, the rats start opening the door on day 3, 4, 5, and by day 12, they get into the arena, look around, same gig as yesterday, and they open the door within a few minutes. So that was exciting to us, and I'm just going to show you what that looks like. So here is day 1. It's sped up. Why is I lost my sound again. Oh well. All right. So what's interesting about this part is that the rat is actually going into the center of arena. And when we saw that, we knew we already had it, because rats don't like to do that. They don't like to be in the center of the arena. So that told us that the rat cared enough. And now what you're going to see is the rat's first opening. This is on day 5, minute 25. The door's going to open, it's going to make a sound, and they're both going to freeze. So that's not a glitch in the video, that's that they freeze, because they didn't expect that sound. The other thing that you see is that the rat that opened follows the rat that was liberated, jumps on him, licks him, and it is always that way. The opener follows the opened. And now, here we are on the final day. You saw the rat being put in. And once he opens the door, what you'll see is that the rats don't pause for a second because they know exactly what's going to happen. They're making an intentional action. So they're no longer surprised by the door falling over and making a sound. And what this tells us is that the rat is making an intentional action and he knows the results of his own action. Okay? So, wonderful. We find out that the rats will open the door. We actually also found out that if the rat can't play with the other rat, he still opens the door. If the rat is actually let out to a different arena, still opens the door. And that's important because the simplest explanation for opening the door is, hey, I want to play with you, right? So we had to see whether that was true. That is not true, and actually, Nobuo Sato confirmed that in a sort of adaptation of our experiment. But I didn't have time to show you those data. So what I'm going to show you now is I'm going to put it into context. So he opens the door. Is that a big deal? Would it happen anyway? Would it happen if there wasn't a trapped rat? And does it happen as fast as it can, or is it slow? And so what I'm going to do is I'm going to frame this in terms of the floor and the ceiling. In other words, what happens if there's no rat involved, and what happens if it's the self rather than another? Okay? So to figure out what the floor is, we just put an empty restrainer in the center, and we see whether they open that. To figure out what the ceiling is, we put the rat in the tube, and then we turn around the door. So now the rat is letting himself out. And he, if he's good, he can stay in the tube. But if he wants to get out, he opens the door. So that's going to tell us that's the fastest that they're going to get out of the tube. And here are the results. So on the left, we have the median latency. So the session lasts 40 minutes, and if there's an empty restrainer, what you see is that they never open. So here's the median latency. This is the percent of rats that open, and we had 8 rats, and 1 rat opened on the 12th day. When we tested him on the 13th day, he did not open, nor did the other rats open on the 13th day. So we had 1 opening out of 8 times 13. 104. And then on the other side, we had these rats that were getting themselves out, and you can see that they very rapidly develop a quick opening. So they're very rapidly going down to a median latency of 0 minutes. That means it takes them, the median of all these rats, this was 16 rats, was instantaneous. And 15 out of the 16 opened for themselves. 1 was chilling. Okay, so that's what the space looks like, and where do the rats fit that are opening for another rat? Well, they fit right there. They're in the middle, but what you see is that they make a switch. First, they look as though they're looking at an empty restrainer, but by the end of the testing sessions, they look as though they're opening for themselves. They're making a switch. All right? So they, and as far as, and most of them are doing this. So they look very much as though they're opening for themselves. Now, if we look at this in a different way, this is 24 rats. Each row is a rat. Each column is a day, 12 days of testing, 24 rats. And if the rat opens once, it gets a 1. And if it opens twice in a row, it gets a 2. And 3 times in a row, it gets a 3, and so on. And then I've color-coded this. So if you open 12 times in a row, which none of these guys did, you get red. But a whole bunch of them get very close to red. And what you can see is that for most of the rats, once they open, that's it. They're going to open. They have switched over. They are no longer making a decision. They've made their decision. And they've probably made their decision because on the first day that they opened, they had a really good time. This is like taking a drug. This is what we do. It gets reinforced. Things that get reinforced are enjoyable. They are rewarding. It's a tautism. So here they are. They switch over into helping. Occasionally, some rats don't do that. These are what we think of as accidental openings. But this kind of thing where they open once, don't open, and then switch over, that's pretty rare. By the time an animal has opened 3 times in a row, the chances that they open on the 4th day is 100%, unless we do something nasty to the rats, which we don't do. Okay, so that's the good. Helping feels good. And it feels good because it's evolutionarily adaptive. It's adaptive to help others in your social setting to live, find food, find shelter, raise young, etc. And so it's going to feel good. We call this, in the human literature, you call that the warm glow of helping. Well, we would argue that the rat gets some version of that warm glow of helping. Recently, Seven Tomek at University of Arizona used our paradigm to pit helping against 2 other substances. One was heroin and one was sugar. And helping lost to heroin but beat sugar. Now, I think that helping could beat heroin depending on the dose. So that is to say that this is going to feel good. It's going to feel good to resolve another's distress. What's the bad? Well, there are a few bad things, but the one I'm going to highlight here in my limited time is the effect of, so in these experiments, it's the same deal. This is 16 rats. Each one of these is 16 rats, 12 days. In this panel, the rats got a saline injection every day. So what is saline? It's pharmacologically inert, but rats are unhappy. It is a stressor. The rats didn't like that. And what you can see is that fewer rats helped, but most of them, they still convert. They still, for the most part, convert. Then we give them a low dose of midazolam, which is a Xanax or Valium type drug. And then we also give them a high dose of midazolam. The high dose of midazolam kills this whole rewarding aspect to helping. Okay? And the low dose does great damage to it. So what that tells you is that you need to spend some affective energy, some emotional energy in order to help. It's going to cost you. It's resource depleting. This is what people refer to as burnout. That's what happens to help, you need to feel. The animals that don't feel anything for the others, they don't help. So the bad is that it's resource depleting, it requires an expenditure of affect, and the other bad thing is that it requires, which I didn't have time to go into, but I'll just mention it, helping requires emotional regulation. In other words, if you see somebody getting a shot, you feel, or even yourself, you feel the pain of that shot, even if the needle doesn't particularly cause any pain. But if you are a physician and you have given X number of shots every day for the last number of years of your training, you no longer feel that their pain. Okay? So physicians have actually trained themselves to downregulate, to not sense the pain that others are having. And this is, and again, this is costly. So the bad thing about helping is that if you're going to do it day in, day out, it's going to cost you. What is the ugly? Well, there are a few ugly things. One is that rats are socially selective about who they help. So all of our work was done with albino rats. And when we initially published this, we published it with albino rats helping their cage mates. And the most common question that we got was, well, what happens if you put in a stranger? And the reason that we got that question over and over again is because in the helping literature and the empathy literature of humans, there is this, I now think, bizarre idea that we help familiar people, but we don't help strangers. Now, this should be news to any charity in the entire world. But there is this strong bias that you don't help strangers. So I asked an undergraduate, David Rogers, this is a bunch of years ago, to do the experiment. I thought it was going to be a simple experiment. I thought they weren't going to help the strangers. I had drunk the Kool-Aid too, and so I said, David, go, design a nice experiment. He comes back. He says, okay, what he wanted to do was he'd have a free rat would see a different trapped rat each day. We had basically a stable of strangers. So each day they saw a different trapped rat. What would they do? So he comes up, and I said, good, great, go do it. And about a month and a half later, he comes up and he says, Peggy, there's no difference. They help the strangers. So we scratched our head about that for a while, and then we realized that these albino rats, as is true for all the laboratory rats, went through a genetic bottleneck back in the early 20th century. So all of them went through the bottleneck that produced Wistar rats, and then there were progressive other bottlenecks that produced the other stocks of rats. So these are they're not genetically identical, but they're very related. And it's as though you've lived on this, in this island for all your life, and everyone you see, they're not your brother or your sister, but they're your fifth cousin. Okay, and they look kind of familiar. And this is an islander, that's not an islander. So we thought, well, what would happen if we tested an albino rat with a rat of a different type? And we chose this very good-looking Long Evans rat, which is a black-caped rat. And we had a couple of conditions. One where the albino rat and the black-caped rat lived together, and one where this albino rat was tested with black-caped rats and had never seen a black-caped rat. And what we found was that if they lived together, he helped the black-caped rat, no problem. But if they'd never seen a black-caped rat, they didn't help him. Along the way, we had a mishap, and I would, I urge you all to embrace those mishaps because they often lead to the most interesting experiments. And the mishap was that in one of our pairs, the black-caped rat died. It was sad, yes. So he died mid-experiment. And mammals, rats, humans, other mammals, really can't live alone. So we paired him up with another albino rat, and then we tested him. 5? I'm done? Oh. Okay, wrap it up. So what happened was, so we paired him with another albino rat and tested him with black-caped rat strangers. And lo and behold, he opened for the black-caped rat strangers. So what that told us was that knowing one black-caped rat allows you to accept any black-caped rat. And that allowed us to do this really cool experiment where we took these albino rats on the day of their birth and we put them into the litter of a black-caped rat, raised them up. They never saw another albino rat in their lives. When they were adults, they helped a black-caped rat. You would expect that. But then what would they do with an albino rat? And the answer was they don't help an albino rat. So that's the helping is socially selective. If the rat is from a familiar strain, they'll help it. If it's from an unfamiliar strain, they won't help it. And so, oops. Skip. So I would just argue. I lost the last little bit, but there is, because helping feels so good, and because part of the issue of helping that feels good is the fact that you did it. So the experiment that I just did, that I wasn't able to just show you, suggests that you can, in fact, feel really good by doing something that you interpret as helpful. But is it helpful to another person? It may or it may not be. And that allows you to misconstrue your own actions in a positive light. And for this reason and for a number of others, Paul Bloom makes a beautiful argument that suggests that you don't actually want to operate out of an emotional empathy. You want to operate out of a more rational compassion. And I am going to take one more moment to illustrate this, just so you think that neurobiology doesn't come in everyday life in very important ways. I just returned from about a 2.5-month illness of my father who died 2 weeks ago. And as he was dying, it was my responsibility to give him pills. And he got to a point where he didn't want them. And he made that very clear. It was very unpleasant for him to swallow the pills. Acting out of empathy, I told my mother, I can't do this anymore. I can't give him the pills. She had been taking pills for so long, she said she could do it. And the question to you is, which one is better? In this situation, my father wasn't going to survive this. He was in hospice. It was okay, but which is the right thing to do? Is it always the right thing to go and feel empathy and to feel for the person, or do you want to do what you think is going to be the rational best thing? Another way to put this, and I really will end, is a friend of mine was telling me he does mostly thyroid surgery. So essentially, he said to me, I cut throats for a living. So a surgeon is imposing damage in order to help you. We're all going to drink this Kool-Aid and say that this surgery is going to help you, but nonetheless, you're going through a step of imposing damage. Empathy is not going to help you in that situation. It's going to stop you from doing it. So I would just argue that it's arguably a better thing to do to dampen down your empathy, go for rational compassion over empathy. And with that, I will stop, and I'll look forward to your questions during the panel.
Thank you. I love the last slide about it's never too early or too late to learn neurobiology. So I know we could have any one of these speakers for the entire time, but we have 3 distinguished speakers. So I'd like to now introduce Dr. Dorothy Kozlowski. She's the interim dean of the College of Science and Health and Vincent DePaul professor in the Department of Biological Sciences at DePaul University. Her teaching and research, of course, focus on neuroscience, and she's interested in how the brain tries to repair itself following traumatic brain injury through neuroplasticity, the triad of our talks tonight. She, her students, and collaborators also study how to enhance brain repair through therapeutic approaches such as physical rehabilitation, stem cell transplantation, or pharmacological agents. Her lab is also interested in understanding why repeat concussions and subconcussions result in long-term neurological deficits. Her research has been published in a variety of journals, and book chapters has been supported by the National Institutes of Health and the United States Department of Defense. Dr. Kozlowski teaches courses in neuroscience in the biology and neuroscience departments, as well as the liberal arts program. She received the Excellence in Teaching Award from DePaul in 2010, and the Educator of the Year Award for the Faculty for Undergraduate Neuroscience in the International Organization in 2011. In addition to teaching and research, Dr. Kozlowski works with DePaul students and the Concussion Legacy Foundation to offer concussion education presentations to youth in the Chicago area. These presentations are designed to teach youth about the symptoms of concussion and how to play sports safely. She is also actively working on bringing attention to the role concussions play in victims of domestic and interpersonal violence. So please join me in welcoming Dr. Dorothy Kozlowski.
Thank you. I really want us to have time for this panel, and this is supposed to be done at 8. So maybe I can just say that everything they talked about is plasticity, to be perfectly honest. So I'm trying to figure out how to do this and give us time to talk. So I'm just going to go quickly through some of this. So as in the triad here, my talk is going to focus on plasticity. And specifically the kind of plasticity that I focus on in my research is looking at plasticity, i.e. how the brain can change itself, how that relates to rehabilitation, specifically following traumatic brain injury. And here at DePaul, this has been done by a whole slew of undergraduate and graduate students, as well as my collaborators who are now currently at the NIH, and then a collaborator that I've had for years. Basically, we were graduate students together who now we still collaborate, and I'm not going to tell you how many years ago, but it was a long time. This work has been funded by DePaul and the Department of Defense. Neuroplasticity. There's lots of definitions for it. This is one of my favorites. And really, it's the idea that our brain has the natural ability to form new connections in order to compensate for injury or to react to changes in your environment, changes with stress, changes with empathy, etc. So I got interested in this basically when I was an undergraduate. And at that time, the idea that plasticity could happen in adults was pretty much poo-pooed. It was really thought that this idea that neuroplasticity is something that really only occurs during development. So I was thinking that, well, this is kind of a cool idea, and maybe I should go study it in medical school, and then I decided, no, I joined this lab, and that got me excited and moved me on to graduate school instead. And it was there that the work that some of the work that I'm going to tell you about today were some of the early demonstrations that plasticity could happen in the adult brain, not just during development. So just to give a quick primer, why am I telling you this? Well, because when we look at plasticity, we actually look at the cells of the nervous system. And when cells talk to each other, a signal arrives in this part of the neuron called the dendrite. It gets sent down the axon as a signal, and it communicates to another cell, right? So there's a number of different places you can look at within the cell to look at plasticity. And the way these circuits happen is that these cells form pathways. Just like if you're going to go from DePaul to O'Hare, you're going to take a certain pathway. If you're going to go back to the loop, you're going to take a certain pathway. And so these pathways are also formed by these different neural connections. What you see here are a variety of pathways using dopamine, some of which may play a role in the rewarding behavior Peggy's rats felt with regards to letting the rat out of the tube. So when we talk about plasticity, we can look at all these parts of the neuron. We can look at these arbors, and when there's more of them, that means there's more opportunities to receive signals. We can look at axon sprouting. So if initially I was going to take the Kennedy to get to O'Hare, but it got clogged, I'm going to go off on the side road and take something else, right? And then synapse formation. So when you get to make that connection, the more synapses you have, the more opportunities there can be for bigger, better connections. So that are key ways to look at individual places where change can happen on cells in the nervous system. But the things that we've looked at is something called cortical map reorganization, which I like to kind of first talk about in a way that is easily understandable to all, and that's Legos, okay? We'll talk about what a cortical map is first, but I'm sorry, this is blurrier, and yes, I stole this image. That was one of those free gifts, but it's a lot blurrier than I thought it was going to be. So, Legos. Pretend each Lego is a neuron, right? In the nervous system, when you're developing these Legos all develop in such a way that they form different things, okay? You can take these Legos, and you can build this house, which is quite cute, but kind of small. Or you can take those same groups of Legos and build this two-story house, right? You can take all those Legos back apart and put them back together and build this windmill, okay? That's one way of thinking about cortical map reorganization. You can take the same cells, but build them in different ways so that the connections and the pathways look different depending on whether you build one of these three houses, okay? That's neuroplasticity, and this is pretty much where we could probably stop so that we have enough time for the panel. But this is the general idea, right? You have the same group of cells, but you can reorganize them and put them in different patterns, okay? But in the brain, we're not building houses and out of Legos, we're building maps. And one of the easiest way to think about a map, we have lots of different maps in our nervous system, but one of the ones that was used initially to discover injury-induced plasticity is this sensory motor system. So at the top there, where's my little, what you have is you have this little strip of brain, and this little strip of brain is called the sensory motor, my gosh, I'm blanking. Anyway, it's it's late. This strip of brain is responsible for your sense of touch and integrating it into movement, okay? So literally what scientists have done over the years is that they've put in little electrodes into these different parts of the brain and said, do you feel this? Where do you feel this? Where do you feel this? And you can map out what the function of these brain areas are. And in this strip, you have a beautiful representation of all the parts of your body and which part of this strip of brain is responsible for the sense of touch in that body part, okay? So now, what happens when you injure that strip, okay? This was work done in the late 90s, early 2000s by Randy Nudo. He took monkeys and looked at that same strip, okay? And he mapped it out. So here's the cortex, the surface of the brain, put in electrodes, mapped out which part was responsible for movement in which body part, and it's color-coded. So here is a 2D map of that. Here's the hand representation in a monkey, and here's the elbow-shoulder representation, okay? Now, if this monkey is given a small stroke, okay, what happens here is that stroke causes that animal to not use their hand as well, okay? It's just like in humans. What happens is that representation in the brain decreases. So there's less Legos, less neurons, okay, responding to that hand because that hand is not really being used anymore, okay? And then the elbow and shoulder takes over. Why? Because the monkey is using elbow and shoulder to do things that the hand was previously doing, okay? Now, so that's an example of injury-induced plasticity, okay? That's correlated or related to behavior. Now, if you take the same monkey with a different monkey with a stroke and then give the animal rehabilitation, so giving it PT, basically. Now what happens is you don't have that cortical reorganization due to the stroke. You don't have a decrease in the hand area and the elbow and shoulder taking over, and the animal actually gets better and actually starts to learn how to use their hand. So this was sort of the first demonstration that, not one of the first, but some of the earliest demonstrations that the adult brain can reorganize following an injury, and that you can affect that reorganization by doing something external, behavioral, giving physical therapy. This was also some of the first demonstrations that physical therapy relies on plasticity. Okay? So this is all fine and dandy, but we were earlier in and around the same time when I was in graduate school, we were not just interested in the area right around the injury in terms of plasticity. We were actually more of a behavioral lab. And at that time, we realized that, well, when we have this injured animal and they're not using their paw as much, they really do rely a lot on this other paw. Okay, just like, when you break your arm or you hurt your arm, you're using the other arm, right? So we were like, I wonder what happens in this injured brain to the area opposite the overused or overcompensating paw, right? So now we're not just looking at plasticity that happens here, but we're looking at plasticity that happens in this opposite hemisphere of the brain, okay? That's the one that's controlling the quote-unquote good limb, okay? And what's cool about this is that this was my PhD advisor, and like I said, we've been working together for a long time. It was one of my collaborators. We saw, remember those dendrites and synapses we were talking about? We saw those cells in the brain changing in a very time-dependent way, and this time-dependent change correlated with the use of that good paw, okay? So we said, well, what happens if you prevent that paw from being used? We put animals in this little cast that prevented, they could still feed themselves, but they couldn't really use their paw to weight bear or to walk. And we forced them to use their good paw. And we said, well, what happens then to this plasticity that we thought was happening, or that we saw previously happening? If it truly is due to the fact that these animals are overusing their good paw, okay, to compensate for the bad paw, then what would you expect to happen here? Is the plasticity going to be there, or is it going to go away? Yeah, it's going to go away, and it did, okay? So this was demonstration that not only can we have plasticity occur, I'm not going to go through all of this, not only can we have plasticity occur just in this area surrounding the injury, okay, but it can also occur in other areas in the injured brain, okay? Long and short, since I only have a few more minutes and I know we want to talk about our panel, this isn't just a rat thing, okay? All these ideas about plasticity and using one arm versus another and the compensation, all of that has actually been translated to humans. So preventing stroke victims from using their quote-unquote good limb, okay, forcing them to use their bad limb, that affects the cortical map in humans as well. So here is basically, not as invasively done, but here's the map of post-stroke humans. So here's the stroke side, the non-stroke side. You give them rehabilitation, just like what was seen in the monkey. Okay? So we were really interested in this, and we said, can this happen following traumatic brain injury? Everything that we've learned about plasticity should apply from stroke to traumatic brain injury. The end result, since I do not have time, is that it does not. The brain following traumatic brain injury is not as plastic as it is following other types of injuries. Okay? So all those things that we saw, cortical map reorganization, increase in dendrites, increase in synapses following stroke, it doesn't happen following traumatic brain injury. Okay? your brain is not as plastic. And because of that, rehabilitation, okay, is not as effective. And when I present this to clinical PTs and clinical groups, they're like, yeah, it's so much harder to rehabilitate our brain injured patients than it is our stroke patients. So the data I didn't get to show you here is when we did a combination of physical therapies in our rats that on their own worked really, really well in stroke, you needed all three of these different kinds of rehabilitation: reaching tasks, exercise, and that forelimb constraint. You need all three in order to show an effect post-brain injury. So plasticity happens, okay? It happens in learning, it happens in stress, it happens following injuries, but just because you think the injured brain can recover following a stroke doesn't mean that those same processes are the same for other diseases like traumatic brain injury, or who knows, for maybe other things like Alzheimer's, Parkinson's, MS, et cetera.